let's bring on Annovis Bio and their founder and CEO, Maria Maccecchini. And there she is. How are we doing today?
I'm doing great. Thank you for having me.
All right. We have Maria's slides, guys. Let's get them up on the screen. There we go. Just get that right click off, Maria, and we'll be good to start. I see a little pop-up menu on your screen. You see that?
No. I have to say next.
Yeah, it would just get you can. All right, that works too. Perfect. All right, the floor is yours.
Okay, so thank you very much for having me. And we have a novel approach to Alzheimer's and Parkinson's disease. What we are really doing is fixing cells that have gotten sluggish. We make them work better and faster. And that is, we are improving the speed by which they work, which is axonal transport. That, as it turns out, works in a number of neurodegenerative diseases. And I'm going to show you today how in our hands it works in phase 2 for Alzheimer's and for Parkinson's patients. So please do read the forward-looking statements. As I just said, this talk mostly will concentrate a little bit on the mechanism and then mostly on the 2 phase 2 studies, one in Alzheimer's and one in Parkinson's disease. As most of you know, we have been attacking neurodegeneration, Alzheimer's specifically, by attacking amyloid, and more newer approaches by attacking tau. And in Parkinson's, we have been attacking alpha-synuclein. While these proteins are toxic, they're all 3 toxic, they do exist in the brain of the Alzheimer's and the Parkinson's patients. All 3 do exist. So it's not that you could just remove one and then the toxicity goes away. You remove one and you still have the toxicity of the other 2. And so, in fact, our drug does lower the levels of all of these proteins. And it does so because nature gave these proteins a regulatory mechanism that is identical. Their translation, which means the way in which they're made, is the same, and the regulation of that translation is the same. And our drug interferes with their translation. This is a picture of how the drug does that. So the green bands is the protein. It's called iron regulatory protein 1. It's a protein that sits in the cytoplasm to which the mRNAs bind. That is the yellow band that is called IRE. So the little loops that I showed you in the previous slide bind to this protein, and when it's bound, the mRNA is not translated. This is true for all of these mRNAs. What our drug does, it keeps it bound. It binds at the interface of the green protein with the yellow mRNA, and it keeps them bound. So there is less translation, therefore there are less toxic proteins for all these proteins. And this is the mechanism. As you can see on top, you see the mRNA with the loop, that's the little red guy. You see the green protein, that is the bivalve protein. That protein binds to the little red guy, and when it's bound, there is no translation. And as I just said, when our drug, which is a small orally available molecule, when our drug is added, it does keep that binding stable, and therefore the mRNAs are not translated. So in a nutshell, what our drug does, it inhibits the translation of a number of neurotoxic aggregating proteins. So what happens when a nerve cell gets injured, when the brain gets injured? The first thing that happens, neurotoxic protein levels are elevated. That's true for A beta, it's true for tau, it's true for alpha-synuclein. What these proteins do, they impair the information flow of the nerve cell. As I was saying, the speed by which information flows is reduced. The nerve cell responds slower. It gets sick, it's attacked by immune system, and eventually it's killed. What our drug does, and that's shown on the right with the blue pristine cascade, you see that it reverses the whole toxic cascade. It lowers the levels of neurotoxic proteins. It increases axonal transport. It has an effect on inflammation. It lowers inflammation, and it protects nerve cells from dying. And by doing so, it actually protects the function that is affected. that function can be motor function in Parkinson's or cognition in Alzheimer's disease. So, not to insult anybody, this is a nerve cell. It has a body in the brain, fingers at the periphery, and an arm that connects the body to the fingers. And the information has to flow up and down that nerve cell, up and down that arm. And these are 3 pictures of that arm. And it shows how the information flows through that arm. So the first picture is a healthy arm. It's a nerve cell that is perfectly healthy. And if you disregard the vertical black lines, you see little black dots that are the carriers of information, and they move smoothly from the left to the right. In the middle, you have an Alzheimer nerve cell. It's a Down syndrome nerve cell. A Down syndrome nerve cell is a sick nerve cell. It is an environment with high amyloid, high tau, and high alpha-synuclein. And if you look at the little black dots that carry the information, they move very haltingly. They stop, they move, they stop. Overall, the speed by which they move through the arm is 50% slower than in a healthy nerve cell. And then at the bottom, it is the exact same Down syndrome nerve cell as in the middle, except it was incubated for 48 hours with ANVS 41. And if you look, you see that the little black dots, again, they flow smoothly from the left to the right. So this is a beautiful example of how our drug restores the information flow of the nerve cell and restores how a nerve cell interacts with the environment and with other nerve cells. Now, in animals, the assumption was and is, well, if we reverse the whole toxic cascade, which we were able to show, well, we should see efficacy. And in fact, in 7 different animal models, we did see efficacy. In 4 animal models of memory and learning, the animals fully recover their memory and their learning, and their cognition is identical to healthy, normal animals. These 4 models were Alzheimer mice, Down syndrome mice, stroke mice, and traumatic brain injury rats. And then we tested it in 2 movement disorders. That's Parkinson's mice and frontotemporal dementia mice. And again, the animals behaved exactly like healthy, normal mice. And finally, we figured, well, if it works in the brain and it works in the body, how about the eye? So we induce glaucoma in rats. And when you do induce glaucoma in rats, you increase the pressure in an eye, the retina dies, and the eye goes blind. If we treat that rat with ANVS 401, the pressure goes up, the retina does not die, and the eye does not go blind. So in animals, we were fully capable of showing the reversal of the toxic cascade leads to efficacy in 7 different animal models. So now we wanted to show it in humans. And so we did a double phase 2 study, 14 Alzheimer patients and 54 Parkinson's patients. This was a double-blind, placebo-controlled biomarker study. In Alzheimer's, we only had 2 doses, placebo and 80 mg. And in Parkinson's, we had 0, 5, 10, 20, 40, and 80 mg. And then, of course, the endpoints were safety, pharmacokinetics, biomarkers, and efficacy. And in fact, we were able to show in humans this time that we were actually reversing the whole cascade and that we met every single endpoint we had in the protocol. The primary and secondary endpoint of safety and pharmacokinetics were met, and the exploratory endpoints of biomarkers and cognition and function were also met. What surprised us in such a short, small study is that, in fact, we saw cognition in Alzheimer patients and motor function in Parkinson's patients. So here is the toxic cascade and the reversal of the toxic cascade. On the left, you see the biomarkers that we measured in Alzheimer patients. It's tau, phosphotau, soluble APP alpha and beta, and A beta 40 and 42. And they all moved exactly the way we were expecting them to move. A beta 42 goes up a little, which was to be expected if a person does better in their Alzheimer's disease. And then in Parkinson's, we saw a decrease in alpha-synuclein, which was also what we expected. Then we looked at inflammation, and all 4 inflammatory markers in Alzheimer's and in Parkinson's, other than YKL-40 in Alzheimer's, were reduced as expected. Not to the same degree in both patient populations, but they were reduced. And finally, we looked at axonal health as measured by neurofilament light, and we looked at synaptic and nerve health as measured by neurogranin. And again, these markers that show that the cell is unhappy were both reduced in both patient populations. So now we got to efficacy. In Alzheimer's, we only had 14 patients. That is not a big study. However, in these 14 patients, the 10 treated patients compared to the placebo patients and compared to baseline, we saw an improvement of 4.4 points from baseline and 3.3 points from placebo, which is a very nice improvement. And then we also measured ways, not just ADAS-Cog, but also ways in Alzheimer's patients. And the ways test measures speed. It measures speed in thinking, speed in recognition, and speed, and that's going to be important in Parkinson's, speed in movement and coordination. And what we saw here again is a 6.6 point improvement over baseline, which is statistically significant, and a 4.9 point improvement over placebo. So in just 14 patients, we saw a nice improvement in ADAS-Cog and in ways from baseline, statistically significant, from placebo, not quite. And then let's go to the Parkinson's patients. In Parkinson's, we measured 0, 5, 10, 20, 40, and 80 mg. And to make it simple, we added up the data of the most and the least significant, so you see the spread. So this is the UPDRS and part 3. Just for your information, part 2, 3, 4, and total UPDRS were statistically significant and showed improvement. Part 1 did not. And here we are showing part 3 in 54 patients. Compared to placebo, 10 and 20 milligrams are highly statistically significant. But if you look at all doses, not just the 10 and 20 that were the best, but all doses, they're also highly statistically significant. Now we move to the ways, and here again, we summarize the data the same way. And again, 10 and 20 are highly statistically significant, and all doses compared to placebo are highly statistically significant. So basically, this study showed us that the best doses to use in phase 3 are 10 and 20 mg, and that the ADAS-Cog for Alzheimer's and the UPDRS for Parkinson's are good endpoints to also use in phase 3. So basically, what we want to do now, or what we are doing, is summarize the data, send it to the FDA, ask for 2 meetings, one for Alzheimer's, one for Parkinson's. In both meetings, we are requesting 2 pivotal studies. So a short Alzheimer's and a long Alzheimer's, a short Parkinson's and a long Parkinson's. And that should then give us enough data to get approval for Alzheimer's and for Parkinson's. We expect to start the first Parkinson's study in maybe April, and the Alzheimer maybe a month later. So right now we are getting ready to send all the information to the FDA to get ready and to get their feedback on phase 3 for Alzheimer's and for Parkinson's. And I'm going to pass it on to Jeff.
Just waiting to see. Can we add Jeff here? There we go. There we go. We got him.
Okay, thank you. All right, thanks, Maria. So this is our patent estate. You can see here we have filed 4 different families of patent applications covering the method of use, covering neurodegenerative diseases, acute brain and nerve injuries, prevention and treatment of diseases, and most recently, last year, we filed for infections. The next slide, the pipeline, we've talked pretty much exclusively about ANVS 401. The other drugs, ANVS 405 is the same mechanism of action, but in acute form, injectable form for acute injuries, and then ANVS 301, a totally different mechanism of action for advanced Alzheimer's. But the focus again is ANVS 401 in AD and PD. And the next slide is a financial snapshot. We completed our IPO just under 2 years ago in January of 2020. We did a follow-on offering this past May, raising enough capital to fund us for another 2 years. And as Maria said, 2 short-term studies, one in Alzheimer's and one in Parkinson's. And likely we would raise additional capital to fund longer-term studies. We also have had the grant funding from the NIH that's funded another trial in Alzheimer's, as well as our chronic toxicology study that we completed last year in animals. We've got about 38% insider ownership, total shares outstanding 8.1 million, a float of 5.6 million, and no debt. The next slide is about the team, Maria and myself, Chen Fang, who is our VP of Research, who joined us about 6 months ago, and Bill Mobley, who's our Chief Scientific Advisor from UC San Diego, who is an expert in axonal transport and Down syndrome. And the next slide is the Scientific Advisory Board, made up of world-renowned experts in neurodegenerative diseases at prestigious universities in the US. And the next slide is, in summary, we have a novel approach to stop both Alzheimer's and Parkinson's. It's the only drug we're aware of that attacks multiple neurotoxic proteins simultaneously. And our phase 2 trial was the first double-blind placebo-controlled study that showed improvement in both Alzheimer's patients in ADAS-Cog and Parkinson's patients in UPDRS, as well as Waze coding in both patient populations. So the successful completion of that study allows us, as Maria mentioned, to plan for our phase 3 trials, which we hope to be kicking off in the early part of 2022. And with that, we can open it up to any questions that we might have.
All right, well, we have like 1 or 2 minutes here. We got a lot of questions. So let me just go arbitrarily here. Are the end of phase 2 meetings scheduled with the FDA for the clinical trial?
One is, the other is not.
Okay.
But the FDA for the one that is said they don't want a meeting, to just ask them in writing, and they're going to give us back an answer in writing.
Okay. Has the approval for Aduhelm impacted Annovis?
That's an excellent question. How much time do I have? In some ways, yes. In some ways, no. Okay? So it depends how you see the future. Do you see the future having Aduhelm, or we can take the Lilly drug, the Roche drug, that also remove plaque, and probably will get approved, and say, well, they exist, they're being used, they remove plaque. What I said in the beginning is we really have more. We do more than just remove amyloid. We also do tau, we do alpha-synuclein, we do TDP-43. Paper was just published, and we'll have a news release on that one of these days. So we actually remove all the proteins that are toxic in the brain. So you could say, well, an antibody for amyloid is very strong just for amyloid, but you may want to have some additional removal of toxicity, which could not be bad. Plus, our drug is very safe, so it wouldn't add to the burden of side effects that you have with antibodies. It really depends how these proteins or these antibodies will be accepted. We can probably do a combination therapy with them, but we can also do a combination therapy with an anti-inflammatory or something else. At some point in our lives, there will be combinations, and I think our drug is very well suited for combinations because it's oral, it's easy to take, it's a pill a day, and it's safe.
All right, Jeff, Maria, thank you both so much for joining us again. We've been on with both of them here. AnnovisBio.com is your website. Everyone, both of you, thank you so much for coming on the Benzinga Small Cap Conference today.
Okay, thank you very much for having us.